right, everyone, thanks for coming. You made it. Final show of the day. So today I'm going to be talking to you about one of the projects that I've been involved with for the past about year and a half. And so this is a project that was funded through the USDA NIFA Beginning Farmer and Rancher Development Program, which is a really excellent program that funds these sort of beginning farmer and rancher projects. And so we used it to showcase some best management practices for veggie growers, small fruit growers, as well as tree fruit growers. And so today I'm going to be talking to you mostly about the data from our tree fruit plots, but we did do some testing at some vegetable plots as well as some small fruit, and so we're still kind of diving into that data, but there's more to come. So our original model plots that were created through this grant we have 4 across the state of Pennsylvania. The first one was on farm at Bedminster Orchards in Bucks County. The second one was in Lehigh County, the third down in Adams County, and then the fourth was actually at the research center right outside State College. And then we do have some of our other vegetable and small fruit plots. We do have some out west, and again, that's some other data that we'll show you a different time. So these plots are 1/3 of an acre large, and the first year in 2015, we actually went ahead and planted sorghum Sudan grass in spring of 2015. And so with the idea that the old apple trees had been taken out of the orchard already, they had been removed, and then with the 2 years in between plantings, we would do cover crops. So the first year we did the sorghum Sudan grass, and then in the second year we did rapeseed. And so we chose these 2 cover crops specifically because they have these allelopathic tendencies that we've been talking about throughout some of these presentations today. And more specifically, these cover crops are able to target nematode populations. And I won't go into the nematode data too much today. We'll be focusing on the soil health. But we did see some really great results from using these cover crops. Essentially, after using 2 years of these cover crops, the nematode populations were 0 across the board. So they're definitely effective, so check them out if you do have some nematode problems. So after those 2 years of cover crops, we went ahead and planted the apple trees in the spring of 2017. And so half of that 1/3 of an acre was planted in Gold Rush apples, and the other half was planted into Crimson Crisp. And both of these were grafted varieties. And they are apple scab resistant. So this past year, in 2017, we decided that we wanted to sample some more apple plots across Pennsylvania to add to our database of information. So in addition to those 4 model plots that I just described to you, we went ahead and added a couple of other plots, mostly actually in the Adams County and Cumberland County regions, and actually one other one in Frederick County in Maryland. And so with those additional plots, we ended up with a total of 28 plots that I'll be sharing the data with you from those. So those additional plots from 2017, we wanted to incorporate some different treatments so that we could look at how those different treatment and management options did affect or potentially affect soil health and those soil health indicators that we're going to talk about today. So the model plots are the Sudan grass rapeseed fescue rotation, which was primarily used for that biofumigation, that allelopathic properties. We also chose some fields that instead of having cover crops rotated in, they had agronomic crops, corn and soybeans, rotated into those 2 years between tree plantings. We also chose some plots that had no to minimal rotation or that had compost added into that rotation. We also chose plots that were just left fallow, so bare, no crop was planted at all. And then we also used a control to make sure that the results that we're seeing are valid and able to be compared. Okay, so let's talk about soil health for a couple minutes here. So as part of our experiment, we decided to go ahead and take soil samples and submit them to the Cornell Soil Health Lab, and we got results from the Cornell Soil Health Assessment. And so all of the data that you're going to see is data that came from the Cornell lab as a part of their soil health assessment. And we took samples in 2015, so before cover crops were planted or incorporated, we went ahead and took soil samples at all of our model plots in 2015, and then in those same plots, we went ahead and collected soil samples again in 2017. So right before the new trees were planted, we took a soil sample. And then in 2017, we also took those additional soil health samples from those other treatments, the agronomic crops, the fallow, the control plots. And so you'll see data from all of those different treatments in a little bit. And so soil health, we've heard a lot about it over the last couple of decades and the last 5 to 10 years in particular. And so by now we should all know that it is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And so when we talk about soil health, we always have to ask ourselves, why do we care? Why should we care? And I think that this graph really sums it up really nicely. So if you look at time down here on the bottom, with increasing time, what we are seeing in these fields are increases in soil carbon. We're seeing increases in aggregation and infiltration, more water and nutrient holding capacity. We're able to see better water and air quality and a higher productivity from those soils. And I think these are all things that we want in our soils. So in general, there are two fundamental ways to assess soil health. So when you are looking at your fields, looking at your farm, you want to be asking yourself, which one of these two approaches do I want to take if I'm going to test for soil health? So the first way you can do that is to take measurements periodically. So that's really more of that first approach that we took, where we took samples in 2015 and 2017. And we want to see, have there been any changes in the soil health due to these management differences? The other way you can do that is to compare measured values to a standard or to reference a soil condition. And so with that, you can take the values that you get from the Cornell Soil Health Assessment and compare that to values that you can find either on a soil survey table. If you go onto the Web Soil Survey, they give you a lot of information about your soils that have already been recorded and are readily available for you to check against. And so when you get the data, you want to be able to use it in a couple different ways, and you choose the way that makes the most sense for you for what you're looking for. So you can make side-by-side comparisons of different soil managements, and that's really what you're going to use to say to yourself, okay, I put in a cover crop, did I even see a difference? Where am I seeing the differences? Is this something that I want to continue doing based on what I've seen in my soil health results? You can also go ahead and take measurements on the same field over time, and that's really more of a check. That's to say, okay, I've decided I like using cover crops. I just want to make sure and monitor it over time to see that it's still working, that everything is still status quo. You can also use it to compare problem areas in your field to other non-problem areas, and you can use that to help diagnose what to do about that problem or how much that problem is really affecting your productivity. And again, you can also just compare those measured values to a reference value. And it's also really important to keep in mind with soil health that you are evaluating your numbers and your results within the context of site and climatic characteristics. And so this, I think, is probably not talked about enough when we talk about soil health. And so the number one thing you need to keep in mind, one of our previous speakers did mention this, you have to compare soil series that are similar, that are the same. And so you really can't be comparing a sandy soil to a clay soil to a silt or a silt loam. They're just going to be, they're fundamentally different. Their soil health is going to be different. So when you're comparing, you have to make sure that those soil textures, those soil types are similar, that they're the same. You also want to make sure that you take note of any signs of erosion. That's going to directly affect some of these soil health indicators that the test is testing for. You want to make sure that you have good notes and a good record of the management history, because what you did 5 years ago on this field or what someone did on this field 5 years ago is going to have a great impact on what's happening today. You want to keep in mind the slope and other topographical features of the field. That's also going to have a big impact. Make sure you remember or note, write it down somewhere where you actually sampled in the field. You'd be surprised how quickly you forget where you actually sampled in a field. And also make sure that you have some records of the climatic information. If it was a dry year, if it was a wet year, you want to make sure that you have that information because, again, you want to be able to use that information to interpret your results. And again, location of any environmentally sensitive areas. If there's a pond in the field, if the field is prone to flooding, anything like that. And so when we talk about soil health indicators specifically, we're talking about 3 separate types. And these 3 separate components are what we use to create a whole picture, to create a complete picture. And so those are physical, biological, and chemical indicators, and we're going to go through all of those today. So with the Cornell Soil Health Assessment that we used in this project, this is the full list of indicators that was tested in the laboratory at the Cornell lab. It's pretty extensive, which is great. So there are 4 different physical indicators that we used: water capacity, surface hardness, and subsurface hardness, and aggregate stability. We also tested for 4 different biological indicators: organic matter, a soil protein index, soil respiration, and active carbon. And then we also did test for 4 different chemical indicators: pH, phosphorus, potassium, and a couple of minor elements. So we're going to jump right in and talk about these indicators in a little bit more detail. So water capacity is exactly what it sounds like. It is how much water your soil can hold. And that's going to be determined by a lot of different factors. And so what you want to keep in mind with water capacity is that it has a lot to do with your soil texture, soil depth, your organic matter, and compaction. So a sandier soil is not able to hold as much water as a clayier soil or even a siltier soil. And that's actually something you can see here on this graph. So on the bottom of this graph is different types of soil textures, and it goes from the coarsest type of soil to the finest, so from sand to clay. And then up here on the y-axis is how many inches of water that soil can absorb per foot. And so you can see that on the left side here with the sandier soils, they're able to hold a lot less water than what we see over here on the clayier side. And it has a lot to do with the size of those soil particles. Sand particles are larger than clay particles, and so the water has more space to infiltrate through a sandier soil, whereas clay particles are smaller, so it takes a little bit more effort, a little bit more time for water to infiltrate. Clayier soils also tend to have more organic matter, and organic matter will just suck that water right up and hold it in your soil for you. So in the Cornell Soil Health Assessment, they were able to do this in the lab. They tested the field capacity and the permanent wilting point, which are the two significant points when we talk about water capacity. So field capacity is how much water your soil can hold at full capacity, and the permanent wilting point, anything below that is, your plants are goners. That's the end of that. They didn't have enough water. And so when we talk about water capacity, we need to talk about what actually is going to affect the ability. So what management is going to have an effect. So tillage is going to prevent organic matter accumulation. And if you use heavy equipment on your wet soils, you can cause compaction, and both of those things are going to reduce the amount of water capacity your soil has. So let's talk about surface and subsurface hardness. And that's exactly what it sounds like. It's how hard your soil is. And so we actually use a field penetrometer, which is what this picture is showing here. And it's a device that you push into the soil, and it is measuring pounds of pressure, actually. So however much pressure you need to put on the device to push it into the soil is what it's recording. And obviously, if it takes more pressure to push it into the soil, the harder your soil is. And so you can end up with a compaction problem when those soil particles are getting squished together. They're getting too close together. So here you can see in this little drawing that there's space in between the soil particles, whereas over here, that space has been greatly reduced. And so again, you'll see compaction with heavy machinery and a lot of tillage. And subsurface hardness in particular can be a problem because it can prevent deep rooting of your crop. And that's going to be really important, especially for nutrient scavenging and when the plant is looking for some extra water. So let's talk a little bit about aggregate stability. Aggregate stability is basically how clumpy your soil is. So within soil, the particles want to sort of clump together and bind together with molecules that you find in the soil. And the better that they are clumped together, the better structure they have overall. And the better structure, the better function. And so the lab actually can test how well those aggregates stick together. And they can do that by either just wetting it and seeing how quickly those particles, those clumps, come apart. They also have a rainfall simulator, which can drop water droplets onto a soil sample and again see how quickly or how easily those soil clumps are able to be broken apart. And this is going to be affected by clay, organic matter, as well as biological activity. And again, we keep harping on tillage here, but tillage is going to prevent organic matter accumulation and disrupt those aggregates, and that fungal hyphae is really important to keeping those aggregates together. And it's important to note that aggregate stability can be one of the earliest indicators of recovery or degradation of soils. So in particular, if you've changed your management style and you want to see if it's working, take a closer look at that aggregate stability. Here's a picture of mulch in a strawberry field to help keep those aggregates together. And then this is actually an example of some pretty bad erosion. You definitely want to avoid that. So when we talk about organic matter, we are talking about all of the living and most recently living material in the soil. So that can be anything from your microbial, dead microbes, dead fungus, plant roots, plant shoots, all of that stuff comes together and becomes organic matter. And organic matter is divided up into these 4 different groups. And we are going to be talking more about this active fraction, the decomposing organic matter, in a little bit, but just organic matter as a whole in general is just a measurement of how much carbon is in your soil. And so that's going to be, again, affected by soil texture, climate, and time. And so what's interesting about how climate and organic matter interact is actually higher temperatures can decrease the amount of organic matter because it's basically speeding it up. It's speeding up the whole process. And similarly with precipitation. And again, so any sort of soil disturbance or destruction of those aggregates, because once you bust open those aggregates, all that organic matter that is sort of hiding inside those aggregates is now exposed and able to be either eaten up by microbes or exposed to the air. Yeah, question?
Can you explain how tillage reduces organic matter?
So that's what I'm talking about.
Could you repeat that question?
Yes, the question is, how does tillage affect organic matter? So organic matter is protected or usually protected within a soil aggregate. And so it's in a complex with the aggregate. So if you open up an aggregate, you're actually exposing organic matter that was previously protected from microbes and from oxygen exposure, basically. And when you bust open that aggregate, now that organic matter is open and ready to be used at that point. Does that make sense? I'll let you think about that. Okay. And so in general, if you are continuously managing for a high organic matter in your soil, you are going to tend to require less farm inputs, which is great. And that soil is also going to be more resilient to drought as well as extreme rainfall. And those things are going to be even more important as time goes by because of climate change and the different types of storms that we're seeing now. And organic matter is a really good indicator of overall soil health. So if you just want to look at one thing in particular on a soil test, organic matter is a great one to say, okay, I'm doing pretty good. When we look at what we call the ACE soil protein index, don't get scared by the name, it's just a way of saying how much organic nitrogen is in your soil. And so organic nitrogen is nitrogen that's going to come from any of your plant matter, so green manures, animal manures. All that stuff is what we call organic nitrogen, as opposed to our inorganic nitrogen, which is our synthetic fertilizers. And organic nitrogen is going to cycle in the nitrogen cycle to become ammonium and nitrate, which the plants use.
What percentage of the OM what percentage of the soil should be OM? What is a good percentage?
That's a good question. So he's asking what percentage of the soil should be organic matter, and that's going to vary a lot. Again, based on soil texture, sandier soils just tend to hold less organic matter, and so their potential for organic matter accumulation is going to be lower than a heavier soil like a clay soil. And so in general, I would say for a sandy soil, if you're around 2%, you're probably doing pretty good. If you are in a more clay soil, probably around 5% is pretty good. And just as a general rule, more is better, but that's not a hard and fast rule. Okay, so the soil protein index is affected by the amount and type of plant residues and your soil amendments. Okay, thank you. So that's just a good way to decide how are my specific amendments doing in my soil health. overall test. Soil respiration, it's exactly what it sounds like. Your soil is actually respiring. It's not the soil, but the microbes themselves. So microbes are like us. When they eat or breathe, they actually will breathe out CO2, and we can measure that in the lab. And it's a way of measuring how many microbes are in your soil and how active those microbes are. And again, that's going to be affected a lot by the organic matter, temperature, moisture, and aeration. So a lot of these things you see over and over again. And it's a good indicator of the biological status of your soil community. So active carbon, this is this active fraction of the organic matter that I was talking to you about a little bit earlier, and that is the direct food source for microbes. So you want to measure that with soil respiration to say, okay, I have microbes, do they have enough food? And this in particular is a good leading indicator of soil health response to your changes. And so you're going to see changes in active carbon sooner than a total organic matter percentage. So if you are interested in testing for active carbon in particular, it's better to indicate what's going on before you will actually see changes in your organic matter percentage. So pH, we all are pretty familiar with pH, so I'll just skip right over that one. pH is important because at different pHs, different plant nutrients are going to be more or less available. So that's why we try to target a specific pH for our soils. And we also did a routine sort of nutrient analysis, which I won't get into that either. So let's jump into some of these results. So for the model plots only, these are the plots that had the cover crops for 2 years. We tested them in 2015 and 2017, and actually overall the soil health scores of most of those model plots didn't change that much from 2015 to 2017, which was curious. It wasn't really what we were expecting at all. The one orchard that we did see change with was the Bedminster Orchard over in Bucks County, and it went from an overall rating of suboptimal, that's how Cornell does their ratings, to excellent from 2015 to 2017. And as far as I can tell at the moment, it did have the largest amount of cover crop biomass, and that could be one of the contributing factors to why it seemed to perform so well and outperform the other model plots. But again, the history of the field is going to be important to say, well, maybe we were just expecting a little too much of those other fields. And actually, a lot of those other fields had very high soil health scores, even in 2015. So when you're starting high, it's hard to really see any increases or any improvements if it's already a very good soil. And so for the other treatments, the other plots that we did look at, these are the indicators that actually we didn't see any changes or any differences between the treatments. And this is just to give you an idea. So with water capacity, surface hardness, aggregate stability, soil respiration, and pH, we didn't see any difference between the treatments. The treatments were the cover crops, the biofumigation crops, the agronomic crops, the compost plots, the fallow plots, and the control plots. So with these specific indicators, we didn't see any differences between those different treatments. So things we did actually see a difference in. We did see a difference in subsurface hardness. And to give you a better idea of what surface versus subsurface means, the surface hardness was at first 6 inches, subsurface was 6 to 18 inches below the surface of the soil. And so what we saw in these treatments in particular is that the compost treatment had a lower subsurface hardness than all of the other treatments, actually except for the agronomic crop treatment. And these are statistically significant differences. So this is based on statistical analysis. And again, I don't really have a very good answer for that, except for perhaps this compost that they were using at some of these sites had higher organic matter. Perhaps it broke down more quickly than the cover crops that we did use in those model plots. We also saw differences in the organic matter percentage. The model plots did have a higher organic matter percentage than both the fallow treatment as well as our control treatment. And again, that's probably because of those extra cover crops, that extra organic matter that was introduced into the soil and allowed to break down for those 2 years. We also saw differences with that soil protein, that organic nitrogen in our soil. So again, the compost treatment showed that it had a higher soil protein index than all the other treatments, actually except for the model plots, the biofumigation. So the compost and the biofumigation showed both were the highest levels of soil protein over the other treatments. And then last but not least is active carbon. And so again, what we saw here was that the compost active carbon was higher than at least the fallow treatment and a tiny bit higher than the other treatments as well.
So what is the unit for soil protein?
That's a good question. I believe it's grams per gram, but I'd have to double check that for you. And so we are seeing a lot of interesting things happening here, and it's important to keep in mind that this is only 1 year of data, and what we would like to do in the future is to hopefully get more data. We'd like to retest these same plots, hopefully a couple years in a row, and see what those long-term changes are. And we'd also like to include more plots in this survey to expand our knowledge and expand our expectations of these different treatments and cropping systems within Pennsylvania. And it's also good to get more background agronomic info so we know where we're starting from to begin with. And that wraps it up for me. If we have any time for questions.
Yeah, take a question or two and we'll call it a day.
Sounds good.
So my scenario is this: it's October, I have 4-foot high Sudan grass. Do I plow it under, or do I just let it winter kill? Which will give me higher organic matter in my soil?
Okay, Art, say that one more time for me.
I got a 4-foot high Sudan grass.
Okay,
Do I want to, it's October,
okay.
do I just want to let it winter kill and don't touch it? Or do I want to plow it under? Which one will give me better organic matter in my soil?
That's a good
Which one's gonna
question.
give you more nematode control?
Right. So Art is asking about,
I'm just concerned
there's-
about organic matter in the soil.
yes, so I always get this messed up. Sorghum Sudan grass, either let it winter kill or plow it under in October, just for organic matter. So if you're going just for organic matter, all you really care about is biomass. So you're going to let it go as long as you possibly can. So I would say you let that winter kill and let it get as much biomass on it as it possibly can. And same for a nematode. So
What I understood is that it need to be cut. And
you do want it to be cut.
In warmer times to actively kill it.
Oh, to let it go.
To-
Yeah,
to
so
do it
in-
like in August, cut it, and within an hour, because of that fumigation, get that incorporated as soon as possible.
Right, so and that's kind of the nice thing about sorghum Sudan grass. Like the previous speaker was saying, biomass is really not a problem with it. So you're going to have a lot of biomass either way. And it's true, to get the true allelopathic potential, you want to cut it up. And we did do that in this study. We used a flail mower to chop it up.
Okay, we better call it an afternoon. Thanks, Megan.
Thank you.